Hello, my name is Dick Shaw. I work for CABI and I'm currently the country director for the UK Centre where we do some of the research on biological control. And I'm also the regional coordinator for invasive species. And I'm very grateful to be able to speak to you, albeit virtually today. I've been working in biological control of weeds for 25 years now. And I'm going to present some of the work that we do here in CABI and start with an introduction of what we're going to be doing. So I'm going to explain what biocontrol is. Some of you know that already, but it will be all starting from the same point. Some success examples, and then what's happening in Europe, basically the lucky accidents that we're benefiting from. And then I'll take you through the process of biological control using Japanese knotweed as a case study. And then I'll talk about some of the progress in Europe, in particular our projects against Himalayan balsam, crasula, floating pennywort, and some other targets that are of note, and come to some conclusions. So here we are. I'm sitting in this office in the middle of that red building above, and that's where we're based within about 61 staff. But in the invasives team, there's about 13 scientists with some technical support and a legendary fellow, Harry Evans. And we host lots of students and training activities, and we have a wide range of expertise specifically in invasive species, but particularly in biological control, of course. And we have, importantly, 2 licensed facilities, quarantine, which allows us to work on almost anything from anywhere in the world in complete safety. We work on a wide range of biological control, of weed biological control programs. The first few at the top there are what I'm going to talk about today, but there's a lot more activity for New Zealand, Australia, and Hawaii, US, other countries. And we also have some mycoherbicide activities, and we do a lot of outreach. So this on the left, the picture is us at Chelsea Flower Show, where the lady in the middle was running the project, and she won a gold medal at Chelsea for informing people about biological control and invasive weeds. We also have a Swiss team. We've got teams all over the world in many countries, but the Swiss team also work in biological control of weeds. And as you'll see from the names here, many of these species are native to Europe and invasive in US and Canada, where much of their work is carried out. And this is the picture I'll explain in a moment. So we have a wide range of targets around the world. So how does an invasive species become invasive? Well, there's whole teams around the world working on predicting invasion. But the one thing that's always a good predictor is whether it's behaved badly elsewhere in the same ecoclimatic range. You can come up with lots of other traits, but there are often as many exceptions as there are proofs to that rule. So that's the one thing. But the one thing that's always true is that they've lost their natural enemies. So in that process, whether that's the answer or not is not always the case, but they've always left their natural enemies behind when they come for a new invasion. And these newly associated species that do attack them don't really do enough of a job. They haven't shifted their host range to include them or attack them at a level enough to keep them under control, hence them being invasive. And the principle of biocontrol is that some of those many insects may be suitable for biological control study. And here we've got an example of a species changing its behavior. I would describe it after introduction, and that's broom, Cytisus sciparius, which is a European species, which grows as a small shrub, sometimes maybe 2 meters tall. And this is what it does in New Zealand when you take those natural enemies away. This is Jane Memmott from Bristol here in New Zealand. And that plant is not, you would not recognize it in Europe or the UK. So that's what happens when you take them away. And as far as biological control is concerned, there's 2 types in general. There's a few more, but the ones I'm going to talk about today for weeds. And the first is inundative or mycoherbicide approach. So basically, you're producing a product that goes in a knapsack sprayer or whatever sort of sprayer to control the species, the invasive weed. And it's normally an expensive product to develop. Therefore, it tends to be used in very high value horticulture like citrus and top fruit, but also in golf courses and amenity land where resistance is an issue. And it's normally used in those environments, but also where a conflict of interest would mean that classical biological control, which I'll talk about in a moment, wouldn't be suitable. I prefer to call it commercial because it's almost always a product that you have to pay for, only a non-chemical solution. There's always a label and a claim and a user and protection and things like that. So that's an approach that would be used for something like Buddleia davidii, the invasive Buddleia species, which there's two invasive species here. Obviously, the Tesco trolleys are not the ones I'm targeting here, but this is an example where everyone, well, gardeners tend to like this species, butterfly bush, it's pretty, whatever. But it's also highly invasive. It comes from China. And if you're trying to run an ancient Victorian railway in the UK, you'll discover that Buddleia is one of your worst enemies because of the structural damage it can carry out, it can cause. And our colleagues in New Zealand have released a Chinese clear pine weevil against this species very successfully. But I think the conflict of interest would probably be too high for European gardeners to accept the damage that would be incurred in their gardens. Classical biocontrol, which I'll spend more time speaking about, uses co-evolved specialist natural enemies from the area of origin of the plant for single release, often single release, to provide sustainable control. So you're going back, you're finding where it came from, you're finding the species that attack it, picking the safest one, or if there are any safe ones at all, otherwise you don't release anything, and then releasing it for permanent control. And that's the end of the story, hopefully. And as far as all biocontrol, including insects, weeds, everything else, there's been over 7,000 introductions of almost 3,000 species around the world, but not many people know very much about it at all. So it's certainly not a new concept. An idea of the scale at which this can work at. Here we have a eucalypt forest in Queensland, Australia. This is a sort of climax community in that particular habitat. And most people would say that climax communities are very hard to invade and they're pretty well secure. But if you climb over the top of it, which is what this plant has done, rubber vine weed, which comes from Madagascar, then you can completely cripple an ecosystem. And some of this was World Heritage quality ecosystem, and it's basically destroyed. And anytime a tree falls, which they're likely to do with that weight on them, nothing replaces it. So effectively, this is what some people would call a novel ecosystem. It's irreversible damage. This is the end of that area. And the scale at which this is operating at 40,000 square kilometers, this is actually bigger than Belgium. And it's the single biggest threat to natural ecosystems in tropical Australia. So a massive invasive species at which you would throw your arms in the air and say, we can't do anything about this. But fortunately, we were able to go back with the Australian support to go back to Madagascar, and to cut a long story short, we found a rust fungus that was specific to the rubber vine and was released, and now rubber vine is coming under massive control as a result of the release of this very prolific rust, Maravalia cryptostegii, and is probably one of the biggest success stories in biocontrol history. Another good example of aquatic weeds and weevils are a perfect combination as far as biological control is concerned. And here we have Salvinia molesta, which many of you may well know. And here we have a lake in Queensland, again, Australia, who have a lot more experience than us in biological control in Europe. And after 14 months, 200 hectares reverted to a small fringe around the edge there and complete control, which is sustained to this day. So this is how dream scenario, silver bullet style biological control can work. And here is the theoretical process. So your population of your invasive weed is the red line, and you have this oscillation around the general trend. So it's, not boom and bust, but, it's population cycles. But after the introduction of your, in this case, weevil as the last example, you end up with this compression and suppression below a line, which is determined as the economic threshold or environmental threshold, whichever threshold you fancy. And over time, you end up with this oscillation that's suppressed below that level, and successful control continues off into the wall and the garden behind me. But let's not just look at theory. Here we have water hyacinth, a very famous species in Europe. And this is an invader that's invaded all over the world, and fortunately has been targeted with biological control widely around the world, apart from Europe. Using in particular this weevil, Neocotina eichhornii, but there are plenty of other agents out there, including a grasshopper most recently released. And here is the data from just Louisiana in the US. And you begin up here at 1 million acres, obviously American measurements, going up to sort of 1.7 million acres, so massive scale invasion. And then you have this suppression right the way back down here, which persists year to year, decade to decade. So the proof of the pudding, the data is there. So this has been around a very long time, over 150 years, and there's a very good code of conduct and principles on which to follow. And there's been over 2,000 releases globally against over 200 weeds and more than 500 biocontrol agents. So pretty good history, and you'd think it would be adopted much more widely in Europe. Well, I suppose if I present the data in a certain way, here is an exponential increase in the use of biological control in Europe for weeds, which is fantastic, exciting to see. But when you put the numbers on, you can see how few have really been released and how early we are in the stage of biological control, given its 100-year or more, 120-year history elsewhere in the world. So this is in stark contrast to biological control of insects, where there's been over 176 species of predator and parasitoid released in the field outside the glasshouse in Europe, in European countries, which when I first heard this was quite revelationary to me. And most of them have had very little pest risk assessment at all, hence how harmonia, the sort of harmonia examples can happen. So this is completely acceptable. Insect against insect, no one seems to care, but insect against weed, everyone gets quite excited and starts getting very concerned, which is an interesting subject to me too. Anyway, we have had a lucky start to weed biocontrol in the EU, and then we wrote a paper not long ago on this particular subject. Unfortunately, they weren't intentional. So we have the only success stories we can point to so far in Europe or in the EU are actually accidental introductions. And Azolla is one that we're particularly involved in. And I think another speaker will cover this a little later, so I'll skip through it quite quickly. But what we have here is a floating fern, the only floating fern in Europe, that's quite invasive, completely blocks water bodies and can have a good sort of 15-centimeter mat on top of the water, can be mistaken for grass, and animals fall into the river or canal. But what also, luckily enough, in the early 20th century rather, the weevil, Sthenopelmus rufinatus, arrived accidentally as a contaminant. And this weevil was no stranger to biological control and has been used very successfully indeed in South Africa. So we were able to find this weevil after its accidental introduction in the 2000s. And since then, we've been rearing over winter to overcome. It's a South American weevil, and it doesn't like our winters and short summers. So what we do is we rear them up over the winter period, and then we redistribute them around the country to control Azolla. If you want to look it up, azollacontrol.com at the bottom there, you can find out more. And this is the sort of results we're having. This is from June, middle of June. And then a month later, you can see it going brown. Many people assume that's going red, and the Azolla is stressed because it's called red water fern. But come a couple of weeks later and you've got complete clearance. Now, this is not normal. Remember the graph I showed you where there was the oscillation around an equilibrium? Well, this is complete devastation for both the weed and the insect. It's a mutually assured destruction, and it's because it's a new association. They haven't evolved together. They've been reintroduced together accidentally because of human intervention, and they have no resistance to each other. But it's a pretty interesting success story, because normally you see it the other way around. A clear water body gets invaded. So a nice story. The other one that's happening as we speak is the ambrosia ragweed biocontrol through the ambrosia beetle, Ophrala communa. And if you want to know more about this, please visit the Cost Smarter website, which has an amazing amount of information in it. But this is an accidental arrival against an invasive weed, which has huge human health issues because of the allergenic pollen. And this is a great, well, it depends what side of the fence you're on. It's a success story because that pollen, we've actually detected reductions in pollen in the air. And presumably significant reductions in the amount of hospitalizations as a result of that pollen. So this is an ongoing success story, but it's not the most specific agent. This is not the one we would have chosen as the most safe beetle to be released. So there's a monitoring program in place to see whether other species can be attacked by this particular insect that arrived, again, accidentally. And the last accidental example I'll give you is the Dactylopius scale against Opuntia, which has been used around the world very effectively. Opuntia is an invasive species in some very fragile habitats in Spain and Portugal. And this scale insect arrived relatively recently and is now spread over 4 million hectares and is doing an amazing job against the Opuntia. There are people who like Opuntia who are less than happy about this, and that's the importance of deciding your targets in advance, and don't get carried away with biocontrol before you've agreed that the population is behind you in a release. But this is ecologically a very successful project, not a project, a very successful introduction. Okay, let's explain how biological control takes place. The normal process, and I'll use Japanese knotweed as the example because I've worked on it for most of my life. This is the plant. Everyone knows Fallopian japonica, I hope. But what you may not know is that there's a massive discrepancy in natural enemies. So in the native range, these pale bars, you can see these species of insect, beetle, larva, Lepidoptera, etc., and pathogens, but very, very few in the UK and indeed Europe. So it has a disproportionate number of natural enemies in its native range, and very few, as I said before, they've been removed, hence giving the plant its advantage. And the insects and pathogens feed across most of the plant, strangely not the roots. There's very few species feeding on the roots, despite looking very carefully for them over a few years. So we have 186 species of Phytophagous arthropod recorded in Japan, actually feeding, not just tourists, things that are proven to feed on the plant. And our job is to bring those back, select the ones that we think are best, bring them back into quarantine, dress up in our fancy suits and negative pressure, HEPA filtered quarantine facility, and test them. And the process we go about is essentially a process of elimination. We start with a literature review. Has it been recorded as a pest on an important crop? Well, let's reject it immediately. And then you have the advantage of field observations. So when you can see your natural enemy feeding on, let's say, in this case, Japanese knotweed, and not feeding on 5 other Polygonaceae that's in the same square meter, then you can make an assumption that it's pretty specific. So you might wish to select that species for further testing. Susceptibility studies are important because it could be that your version of the species is not the same as the version you've just found in the field, the Japanese knotweed in this case. So you need to make sure that your invader is susceptible, and you'll see this example later with the Himalayan balsam project. And then we start the expensive stuff, which is the host range testing and quarantine. So that's the bit you want to minimize as far as possible. And you end up eliminating, in our case, 185 species and ending up with 1. Which is the Aphelara, which I'll talk about in a second. And how do you go around testing these species? Well, you start off testing the most closely related species, members in the same genus, and then you work out to subtribe using phylogeny, plant phylogeny, and now molecular phylogeny to help you not test the 2,000 species that you have in the field. Plant species, you want to test a limited number, although we did test around 90 species with the knotweed project. And we ended up with this species, Aphelara itadori. Itadori is Japanese for Japanese knotweed, so it's named after its host, which is nice. Very small organism, 2 millimeters long, and you can just about see the eggs with the naked eye. It's the nymphs that do the job, not just the adults. The adults just feed and lay eggs. The nymphs build up numbers and feed more intensely. And the host range testing, I'm going to compress effectively 5 years of research into one slide, but this is multiple choice oviposition. So it's seeing in a cage with a load of different species, where did the adults lay their eggs given a choice? And they obviously laid many on Fallopian japonica, our target, the Bohemica hybrid, and various other invasive fallopias or non-native fallopias. And then obviously they're not very popular down here. And then you have these eggs to your right on rhubarb and buckwheat and Fallopian convolvulus and other polygonums. But the ones to the right of the red line could not sustain development to adult. So they're not hosts, they're accidental eggs. And we are talking about effectively a very specific agent indeed. So how far do we get? Well, it was a very long project. We tested over 87 species. We showed impact in the lab. Sometimes it's hard to keep the plant alive because the psyllids outbreak and kill them. We showed its significant reduction on rhizome weight in field tests, in caged field tests. And then we began our release program. And that's when the excitement started to curtail, because we had trouble establishing it. And this is quite common in biological control. We had trouble establishing and trouble having impact. And overwintering was successful on a few years, but in reality, this is not a success story yet. And we had to go back and reconsider what was going on. Part of the problem could be that they had been in the Japanese summer for 5 years and 130, 40 generations of having no winter. So they've been conditioned for the lab rather than the field. So we did some climate. There's also an issue that it could be climate matching, because we collected down here in Mount Aso in Kyushu, which is subtropical, but up a mountain it was much cooler. But this climate mapping, which Corinne Pratt did for us, shows that we should be much further north. So we went back again in last year, Daisuke and myself, and we collected a new strain from Murakami. And this has a very interesting impact that we don't see with our current strain, and is highly damaging in the field. It creates this curling, almost gall-like structure, and there's your nymph feeding just inside it. So we're quite excited about the new opportunities. And we're currently at a point where the Netherlands is now considering releasing this strain against the Bohemica, which is what it prefers over our Japonica. But I can talk to anyone at length about this afterwards, because I'm running a little tight on time. The other thing we found was a leaf spot. So the only Japanese knotweed you can see, the only reason you can see Japanese knotweed in this multiple species picture here is because of the leaf spot. There's big splodges of brown and red are only on Japanese knotweed and not on any other species, including a couple of Polygonaceae plants in there. So that leaf spot, we did all the same testing that we did for the psyllid, and we ended up rejecting it because it attacked a native species. But what we discovered was that a single mating type of that Mycosphaerella could not generate the spore that would infect the non-target, and therefore it could potentially go through a sprayer and be used as a mycoherbicide. So we've just had a European patent granted for that particular mycoherbicidal product, and we're in the process of doing field trials to see whether this proof of concept, to see whether this is a feasible solution for knotweed in the future. So watch this space. Okay, on to a few other species in my last 10 minutes. We have Himalayan balsam, Impatiens glandulifera, a major invader that attacks, invades waterways and riverbanks and meadows, and is pretty difficult to control. Even though it's an annual with a limited seed life, lots of people pull it or balsam bash. They're called balsam bashing. This is one of those species that you need to deal on a whole catchment scale. You can't just deal with your 100 meters because someone 100 meters up the road, up the river, has got another plant that's throwing hundreds of seeds into the river. It can be pretty amazing in its invasion and completely exclusive. And obviously with 5,000 to 6,000 seeds per meter square, it's a great threat to very vulnerable habitats. So the native range is up here in the Himalayan foothills, and that's where we went for survey work. Rob Tanner, who you'll be hearing from later, did much of this work and got his PhD on the subject and is a world expert. So if you've got any questions, I'm going to point you to him. But part of that process was collecting lots of really amazing organisms and rejecting them all because they weren't specific enough. But we did end up with this amazing rust, which hits the seedlings at one stage and then infects the plant and the leaves. Popsinia comarovivar glandulifera. It was a new to science. We had to name the species and write the paper before we could write the PRA. And this has since subsequently, after 75 species testing, and I should explain with pathogens, the host range testing is a little different with insects. Insects, you give them the food, no choice, they feed or they don't feed. If they don't feed and they die, you know they're never going to feed in the field. That's the extreme test of no choice testing. With pathogens, you need to give them massive inoculum, perfect temperature, 48 hours of due period, and monitor them for twice as long as the target species. And if you don't get infection in those conditions, you're never going to get infection in the field. So that's how precautionary host range testing is made with pathogens. So we had the releases go ahead, and what we discovered was there's a variability in the susceptibility of the plant to the pathogen. So it looks like there's been multiple introductions, and it's a sexually reproducing species that's been separated from its pathogen for a long time. So what we have here is very different infection levels. And what we've realized is that our rust samples that we have are not necessarily optimized for all the sites. So when you choose a site for a Himalayan balsam release, we have to do a susceptibility test first to find out whether that population is susceptible and whether it's worth releasing that particular rust. Daisuke Kurose did a lot of molecular work in our team, and he revealed that there was probably 3 major varieties of Himalayan balsam, and one of them is very likely to have come from the Wangat Valley in Kashmir. And that's where we need to go to get that dream rust that we're missing. And this has been the life's work of our colleague Carol Ellison, who sadly passed away earlier this year to cancer. But we hope to be able to finish her legacy and find that rust from Kashmir and get it released. Okay, on to Crassula helmsii, an invader from Australia. And Sonalvaria is running this project. After surveying in Australia, we found a mite, an incredibly small eriophyid mite. So anyone that's worked with Crassula knows how small the plant is. This is the Crassula, and this is the tiny tip of Crassula, and these are all the mites. So this is an absolute beast to work with, and Sonal did an amazing job just completing her PhD on this. I won't take you through the whole life cycle, but it breeds and survives in the buds and causes this stunting that you can see here, the red coloration. And it was released. It's the first ever. So the psyllid was the first ever insect to be released against the weed in European Union, and this was the first mite to be released against weed, and the balsam was the first rust to be released against the weed. So it was a new species to science again. We had to write that paper. That was another challenge. And after release, you're seeing this is actually in the field. You're seeing this sort of beginning of a stunting of the plant, and this is in the polytunnel, and you can see it's not doing very well at all with the mite in play. So this is very, very early days, but it looks like we're starting to get establishment. We've been plagued by flooding and other things like that, but this is got great potential. On to floating pennywort, a very serious invader, particularly for the UK and Netherlands, Germany as well, and I'm sure Belgium as well. And here we have a weevil. So this is a weed that cost the UK economy 25 million and very, very much more in the Netherlands. Manual removal is favoured. We went back to Argentina working with our colleague Willie, our friend Willie from Fuede in Argentina, and you can see this sort of stunting and ephemeral patches of hydrocotyledon. And the best agent again was a weevil, floating weed and weevils, remember? And this is Listranotus elongatus. And it has a nice life cycle. Adults chew the surface, mating, lays eggs, many eggs sometimes in the petiole of the plant, which mine down the stolon up to 30-35 centimeters, and then pupate and re-emerge to continue. They can survive winter, they can survive freezing, and they get 3 generations of the native range and possibly 2 in the UK. We've finished host range testing. This is Jami Jador's project, and 74 species tested. A lot of issues with export and ABS and access and benefit sharing, Nagoya and things like that. But we got all the work done, and it's currently being reviewed by the UK authorities, and we're hopeful for permission to release in 2021. And obviously, this will be of great interest to our European colleagues in mainland Europe, because this is a weevil versus a floating weed. The only issue we had was some host range issues with the native hydrocotyledon and apium, a very rare plant in the UK, not so rare in mainland Europe. And this is a choice test to show, even though we had feeding and you could get just about through to adult and very few numbers, in a more realistic, in no choice testing, I should say, in a more realistic setup, here we have the mixed plants, hydrocotyledon around the outside, apium in the middle, hydrocotyledon around the outside, and hydrocotyledon vulgaris, the native, in the inside. After 10 mating pairs, 2 weeks later, all you can see is the apium left. The hydrocotyledon is absolutely hammered. And again, with the hydrocotyledon vulgaris is pretty healthy in the middle, no damage. And so it just shows you that this thing really, really does like floating pennywort given a choice. I won't cover acacia because you're going to be seeing a presentation from the Marchantes very shortly, who I'm very pleased with the work they've done. It's amazing, and you'll see some really good results. It's a long burn because it's a tree, but yeah, you'll see anyway. So there's plenty of opportunities out there, basically, in Europe. And this is a paper we wrote a long time ago, 2006, which we're rewriting at the moment to update it. But it's important to look at that EU red list of species that are of European concern. And there's a great collection of weeds in there that have perfect or very, very great potential for biological control. There's some that have been worked on already. Don't forget water hyacinth. Well, I'll come back to that at the end. And then we have a few agents not quite yet released, but they have potential, big targets that have major impacts, that have agents already identified, and others that have been released. So it would be mad not to look at these. That's basically the message I'm going to leave you with at the end. Ludwigia is one of the great importance to, in particular, France and wetlands of France. And the Americans and Willie, the guy in Argentina, has been working on this for the Americans. And there's two great-looking agents and others, including this Leothrips, this thrip species, which causes pretty severe damage and is highly host-specific to the target group of Ludwigias. So we've been trying to develop this project for Europe, in particular France, for 8 years, and we haven't really got anywhere yet, but it's on the list and there's a potential biocontrol agent. Why would you not consider researching that? So what's the alternative of not doing this? Well, with invasive species, doing nothing is not a low risk option. It gets worse and worse and worse. So not considering all the tools could be an expensive option. And remember, we went back to the water hyacinth example, and I showed you the graph and what had happened with the weevil. Well, this is what's happening in Europe, where people are basically saying, well, we can't consider biological control because we don't like the sound of it, it's dangerous or whatever. And what we'll do is we'll do mechanical removal. Well, this is the one river in Spain, the Guadiana River, where they've so far spent 23 million euros pulling it out of a river. And then, funnily enough, it came back and they spent another 5 million pulling out of the river. Now, this has pretty heavy ecological consequences. This bank is not as healthy as it perhaps should be, but more importantly, is wasting that money. 5% of that could have funded an assessment to get a cold-tolerant Neocotinus strain to see whether that might be suitable in quarantine. 23 million would have funded all of the species that were in that list of the paper I showed you 3 slides ago. So this is what I consider to be unwise spend of money. Anyway, I won't get myself into trouble because it's being recorded. Anyway, I can conclude now to say that invasives are a serious issue. Classical biocontrol can, not always, but can solve the problem, and it's worth investing in to see whether it's a potential tool in the armory. It's not quick, it's not straightforward, but it's a long game and will provide a sustainable solution if successful. There's little awareness around and a lot of misunderstanding as well as lack of understanding over the technique in Europe, but it is on the rise and we're starting to get some traction, and you'll see another couple of examples shortly. Another thing I always say is a biocontrol project is there's no commitment to release. It's another piece of research looking at an alternative control measure, and a dossier is what comes out at the end for someone to assess, and a pest risk assessment if necessary. So it's not a one-way road. It's a really great job, though, I must say, and you get to see some amazing people. So I'm going to leave you with some thanks from our donors and supporters, and the many, many people that have been involved in any of those projects. And if you do want to keep in touch, please do. A Twitter page, and we have a blog and everything else. And I'll leave it now for questions, and I think I'm just a couple of minutes over. So thank you very much for your time, and I look forward to talking some more and hopefully seeing more biological control of weeds in the European Union. Thank you.